So thanks everybody for coming. We're going to go through our session here. It's going to start with a slideshow. We're going to have opportunities for each of you to participate via the chat, and then also you can raise your hand if you have questions. We'll try to get to everyone's questions as we can, and then we'll break out into our breakout rooms where you can work with the mentors directly. So today we're going to be meeting an undergraduate student. Her name is Charlotte, and she goes by Charlie Andreasen. She's a MechE, mechanical engineering undergraduate student, researcher in Dr. Rebecca Taylor's lab. She's also studying education or public policy. So I guess I'll go ahead and introduce myself. I'm Kristen Lavery. So you may have seen me if you ever participated in our Gelfand outreach programs. And I've taught a few of our Gelfand outreach classes. I've been with the Leonard Gelfand Center for about 8 years now. My background's in elementary education. I enjoy do-it-yourself projects, dogs, and learning new things. So next up, we have our group, our coordination group. And our first member of that group is Dr. Vicki Webster Wood. She's an assistant professor in mechanical engineering. So Vicki, if you want to introduce yourself.
Excellent. Yes. Hi, everybody. I'm Dr. Webster Wood. I am in my 3rd year as being a member of the faculty here at Carnegie Mellon. And my research is in biohybrid and organic robotics. So we're using living materials to build functioning robots. Because of that, my lab houses 2 saltwater aquariums, and we keep our own sea slugs to study for research. I also play trumpet, mountain dulcimer, and love to quilt. I also love dogs. I have 2 dogs. And fun tidbit, when I was in high school, my very first car came to me with a box of engine parts and did not run. And the agreement was that I had to learn to repair it before I could drive, which was a great learning experience, and you know, I think probably contributed to me being a mechanical engineer. So I'm happy to be here, and it's exciting to see everybody.
All right, thank you, Vicki. We're happy to have you here. So next, we have a graduate student researcher. He's Saul Schaefer. He is a PhD student in Dr. Webster Wood's biohybrid and organic robotics group. So he is a part of the Borg lab as well. Saul, would you like to take a moment and introduce yourself?
Sure. Hello, everybody. So my name's Saul, and as you can see in this wonderful picture, I've got this beautiful handmade CMU blanket, which I sit on nearly every day. And I love going biking in Shenley Park and making my own hummus at home, something I enjoy doing. And then for this Halloween, I'm going to be Avatar Aang. So I'm looking forward to dressing up for that. Should be a lot of fun.
Very cool. Thanks, Saul. And then the last member of our group is an undergraduate student, Allison Rojas. Allison, if you want to take a moment and introduce yourself.
Sorry, I was muted. Hi, I'm Allison. Nice to meet everybody. I'm also in the biohybrid and organic robotics group with Vicki and Saul. And a few things about me is that I helped with CMU CS Academy, which is a graphics-based computer science curriculum. So we try to give free CS education for high school students. And then something else is that I illustrated my own comic book about an alpaca. So I really love like working with watercolors and like drawing and painting. And I have, oh, and I probably should have said, yeah, I'm also a mechanical engineering student. But yeah, oh, yay, I see somebody really likes CMU CS Academy. So that's really exciting. I'm glad you enjoy it. And then I recently took up dancing, like improv dancing. So it's been really fun too. And I'm really happy to be here and meet everybody.
Fun stuff. Thank you, Allison. And lastly, we do have our Road to Research mentors, and all of our mentors for this program are CMU undergraduate or graduate students. So if you all would sort of like to take a moment to say hi to the group, you're welcome to share your video for just a moment or say hi via the chat.
Hey, I'm Julian. I'm a Carnegie Mellon mechanical engineering graduate student working on robots. I'll keep it short because there's a lot of us.
Thanks, Julian. Happy to have you. How about Maddie? Would you like to introduce yourself?
Hi, I'm Maddie. I'm a chemical and biomedical engineering student, and my research is currently trying to find a way to develop a test for Parkinson's disease. So,
hi everyone, I'm Mike. I'm a electrical and computer engineering PhD student, and I also study the brain by using artificial intelligence to understand the function.
Hi everyone, I'm Sharfin. I'm an undergrad mechanical engineering student, and my research project is to work on this guy here, which is supposed to be like a little walker, and hopefully it'll be the size of little Lego guys soon enough. So yeah.
Perfect. Thank you all. Okay, so right now we are going to have our first poll. So this will be where you all can participate. So our first question here, let me get to our slide. All right, so mechanical engineering involves which of the following? So please go ahead and vote for your answer. And these are anonymous, so we won't know what you say, but we like to see that you all are participating. Okay. So for our poll responses, the answer is actually all of the above. So mechanical engineering uses skills and ideas from design, mathematics, production, physics, and more. So this is my little word cloud before we get started. And these are just a bunch of terms surrounding mechanical engineering. So you may hear some of them today, you might not, but I think it will be a fun session, and hopefully you might be interested in learning more about mechanical engineering after today's session. So without further ado, we're going to go ahead and meet our researcher today, Charlie. So Charlie, thank you for coming today.
Hi guys, I'm Charlie. I'm a senior at CMU, and like Kristen said, I'm studying mechanical engineering and engineering and public policy. So I'm from Wisconsin, but I'm actually calling in today from Chicago. So I'm living here this year doing school remotely. I'm sure a lot of you are doing similar things with remote classes right now. In my free time, I like to play viola and violin and upright bass. And I also like a lot of sports, and I play on the CMU's women's ultimate frisbee team. And I'm also currently living with 3 cats, and they're shown on this slide. Sammy is the one in the chair, Rue is the one on the shelf, and then Boots is the one on the blanket there.
Great to have some animal company, especially now.
Oh yeah, yeah, they're very entertaining.
I can only imagine what having 3 cats in the house might be like, and trying to do all of your student work at the same time.
Yeah, I wouldn't be surprised if at some point you guys hear them today, because they'll come in and just meow at me if they're hungry or want attention.
Be on the lookout for any feline visitors to the program today. So we're going to go ahead into our next poll. So this is a process in which the following 7 steps are followed in order. So let me share our poll here. Give me 1 second. Okay, so this is a process in which the following 7 steps are followed. Number 1, ask a question. 2, do background research. 3, construct a hypothesis. 4, test your hypothesis. 5, analyze your data. 6, draw a conclusion. And 7, share your results. I'll give everyone
Kristen, I think some students are saying they can't see the poll.
I am seeing that. So you should have a separate pop-up window. And that's where your poll should be. If you're accessing from your browser alone, potentially you might have to allow pop-ups. Okay, so I'm going to end our polling now and share our results. So it is indeed the scientific method, and we're going to talk about that in more detail, and we're going to talk about Charlie's first experience of using the scientific method. So Charlie, you had your first experience with the scientific method in middle school. Can you tell us about that experience?
Yeah, so when I was in middle school, there were some new policies about how middle school lunches had to be healthier, and so they had to have fruit and a vegetable and a milk, and it's all good stuff to stay healthy, but they made the lunches a lot bigger, and so there was a lot of extra food being thrown away. And so me and one of my teachers were kind of frustrated by how much wasted food there was. So we decided that we should start composting. But the principal at our school didn't really want us to start composting, and he thought there would be, a lot of like bad smells, and it would attract bugs and animals. And so we basically followed the scientific method to do some research into why we should start composting. And so we, like every day during lunch, we would collect all of the food waste in these big buckets, and we were weighing it to see how much was actually being thrown out each day. And then we also did a lot of reading up on composting and the benefits and what you need to do to prevent it from smelling or attracting bugs and animals. And so then we basically submitted this report to him and just said, based on all of this, we think we should start composting. And he did let us start doing it after that.
Great, that's awesome. And I see some of our friends here today know people or do composting as well. So I'm certain you can find out some more information about that too. And right now, we are going to ask you. So who has done composting before? I like seeing these results come in. So our options are A, I have composted before, B, I have not composted before, or C, ooh, worms. Okay, it looks like most of us have voted. And half of us that responded have composted before. So moving on to Charlie's middle school interests. So Road to Research student friends, when Charlie was in middle school, she wasn't that interested in science. So what subjects were you interested in middle school?
Yeah, I thought science was kind of boring, and really liked pretty much everything else. So I liked math and reading and gym class and orchestra, but science was just not my thing.
All right. So what actually changed your mind about science?
Yeah, so when I was in 8th grade, my science teacher told me that I should read this book, The Immortal Life of Henrietta Lacks. And it was super interesting. And they talked about all these scientific discoveries, and then sort of the ethics of science as well. And there's so many ethical issues in science, like especially new things where they're like, starting cloning and all like genetic mutations and all of that. Like there's a lot of ethical issues involved. And so this book really got into not just like the science, but the ethics. And so once I read it, I was like, I need to learn more about science. Like this is actually really interesting.
Definitely. Raise your hand if you have ever, there's a movie now about the immortal life of Henrietta Lacks. Raise your hand if you've ever seen the movie or read the book. And in order for you to raise your hand, all you need to do is go to the participants list. And then from there, you'll be able to raise your hand. So a few of our friends have heard about it. That's always good to see. It's also something that I'm sure Charlie would recommend that you read or even watch.
Yeah, for sure. I haven't seen the movie, but I heard it was good and the book is definitely worth reading.
Yeah. I haven't read the book. I have seen the movie. Usually the book is better. Okay. So tell me about this 11th grade research project. It focused on elementary science education. And can you tell us something about that? A little bit more about that?
Yeah, so when I was in high school, the public schools in my hometown in Wisconsin were cutting all of their elementary and middle school science programs. And so at this point, I was really interested in science and was frustrated that they were cutting these programs like where you do experiments in classes. And they were instead focusing just on like math and reading. So students would get these reading packets, and instead of doing a science experiment, you would just read about the science. And so I was frustrated by this because I was really interested in science and thought more people should be interested in science. And just having a reading packet isn't really the same as doing experiments. So
No, it's not.
so I developed these different teaching methods. So either like a hands-on lab, just these kind of flashy demonstrations, or a reading packet all on the same topic of sound waves and physics. And so then I went into a bunch of different classes and taught these lessons and then gave the students tests right afterwards. Don't worry, it wasn't too hard, but just wanted to see if they had learned anything and which way worked the best for people's interest in science and also like how much they were learning. And so we found that these labs actually were more beneficial to students, like they learned more, they retained more of the knowledge, and they actually liked it a lot more than just doing a reading packet. And so we did this, and we're hoping that more of the teachers would start doing these labs again instead of just giving out reading packets.
Exactly. So that's why we're doing hands-on projects and activities with this program as well, because personally, I like to be able to see and also do things with my hands. So you will get to do that today. Raise your hand if you think it's more fun to have hands-on activities versus just having a packet in school.
People.
Overwhelming response on that one. Nice. That's great to hear. So you were involved in this work with an elementary school. Were you involved in any other groups or student organizations or clubs, Charlie?
Yeah, so in high school, I was interested in science, but I wasn't really sold on like going into engineering or doing more science things. So I played sports, I played in the orchestra. I was involved in all of our like school plays and musicals. But then I also was like on the robotics team and ran our school's physics club.
That's fun. So how did you make physics fun? Why did you enjoy science at that point?
Yeah, so our physics club was really about trying to show people how much fun science really was. Because when you're in the classroom, a lot of it is just doing math and derivations, and it's not super exciting. So our physics club was, you know, trying to show that physics can be used to do a lot of different like fun, exciting things as well. So we did things like calculating the speed of light by melting marshmallows in a microwave. And we did a school-wide egg drop competition. We got like 30 people on rollerblades and then learned about momentum by pushing off of each other and calculating velocities. And then you could calculate your partner's mass based on your mass. We built a giant catapult and launched things off the roof of our school. And because we knew the physics involved, we calculated where this basketball would fall and then put a bucket there to catch it. So using physics, we were able to do all of these fun experiments and like show that we knew how all of these calculations would work and turn out.
That's awesome. It sounds like you had a lot of opportunities to really get involved with physics. And that's great to hear. Hopefully our middle school participants will be able to be super involved like you were in high school as well. So after high school, you of course decided to come to CMU. And what did you originally want to major in here at CMU?
Originally, I wanted to major in biomedical engineering. And so I was going to do a double major with biomedical engineering and mechanical engineering because I really wanted to study medical devices. But then as I was taking some more classes, I really got interested in engineering and public policy because you look at a lot of the ways that policies affect, you know, the engineering process and standards, and like how people interact with technology has to influence how you design technology. And so I thought it was super interesting. So I ended up switching from biomedical engineering to engineering and public policy. But as you'll hear soon from my research, I'm still interested in these medical technologies.
Yes, exactly. And so while we're sort of touching a lot on the mechanical engineering component of it today, what does engineering and public policy mean to you?
Yeah, so that's just a lot of the, you know, how people interact with technology, how that's going to influence what we're doing as engineers, and things like risk management, which typically when you're just an engineer, you may only think about like the cost of your design or whatever constraints you have, whereas someone who's more involved with policy might think about managing the risks involved and the way that people will actually use different things. and how policies need to be designed to like keep up with technology and also manage these risks. And so there's just a lot of different interplay between different topics that just engineers might not typically think about.
Exactly. And it looks like we have a question here. So Peter's asking, what sort of things does a biomedical engineer do? Dr. Webster Wood, would you like to answer this one?
Sure, I can chime in. So I am primarily a professor in mechanical engineering, but I actually also have a courtesy appointment in the Department of Biomedical Engineering. And my research also kind of overlaps both fields, which is something that you see really commonly at Carnegie Mellon. And so biomedical engineering is a pretty interdisciplinary field of engineering where the primary goal is to understand how biological tissues, how health is influenced by all of these different external factors, and how to repair or treat those tissues. So the big idea is, you know, making people healthier, making people's lives better, being able to fix a lot of these issues or injuries that might occur. And so biomedical engineers will do everything from studying the brain and how the brain works and send signals around how to repair bones you know, there's these really fancy knee replacements that people can get now as they get older. My grandmother recently had her knee replaced, and that's all, you know, through biomedical engineering, figuring out how to integrate these mechanical components with your body and with biology. And it even goes beyond that, as you'll see with Charlie's research. It's very interdisciplinary into the BME space. BME is short for biomedical engineering, where you can build with biology. So you're going to see Charlie talk about building with DNA. In my lab, we build with living muscle cells. And so you can use that to build kind of these devices that can deliver medicine or perform these little tiny repairs inside the body. Hopefully that was a sufficient answer.
Okay, thank you, Dr. Webster Wood. So we're gonna let Charlie drive here. So Charlie's gonna be sharing her screen for the rest of our presentation here right now.
Yeah, so I think I am sharing here now.
Oh, yep, you're good.
Okay, awesome.
Great.
Yeah, so now we're gonna just dive a little bit into what my research is actually on. Okay, I guess we're starting with the poll. So Kristen, do you have that?
Yes, we are.
Awesome.
So here is our next poll. And the question is, what is the function of DNA? Is it A, to provide energy, B, to store information, C, to regulate pH, or D, all of the above? So we'll give you another 45 seconds to answer or so. All right, I'm just going to go ahead and end it a little bit early here because it looks like we have everyone participating that's going to participate. So our polling results show that D is the answer. Charlie, is that the correct answer?
No, so a lot of people guess that it's all of the above, but actually DNA is what stores information in our cells. So all of the information about what makes you, you is stored in your DNA.
And Charlie, what is DNA?
So DNA, or deoxyribonucleic acid, is a it's a molecule in your cells that it's an information storer, and we'll get a little bit more into the structure in a minute. Oh, how are these slides working? Is this the one we just did? No. Okay, sorry.
It looks like we have a duplicate slide though.
Yeah. Yeah.
One
so-
extra jumped in.
Here though.
Okay. So do we want to go to our second poll now regarding DNA? Okay. So now here's another question for you all. Where in the cell is DNA located? I'll give you about 20 more seconds to respond.
All right. It looks like most people said nucleus, and that is correct. So DNA is an information storer, and it's located in the nucleus of the cell.
Okay. So Charlie, tell us a little bit more about the structure of DNA itself.
Yeah, so DNA is made up of nucleotides, which are basically we have a base and a sugar phosphate backbone that holds all of these bases together. So each nucleotide is made up of one of these sugars and phosphates in the backbone held together or bonded to a base. And then our entire strand is made up a whole bunch of these nucleotides all stuck together. And so what we have here is our bases that bind together. So there are 4 different bases: adenine, thymine, guanine, and cytosine. And our cytosine and guanine bind together, and the adenine and thymine bind together. So when we have these bases binding, they actually form this double helix structure. And I'm sure a lot of you have heard about that in science class before, the double helix structure of DNA. And so basically it's just a twisted ladder, and each rung of the ladder is one of these base pairs, either an AT base pair or a CG base pair.
And raise your hand if you have learned about DNA yet in school. Okay, it looks like some of you have learned about DNA. Cool. You're going to learn more about DNA today.
All right, so I don't think we have these as a poll, but if you want to just throw it in the chat quick, does anyone remember what the 4 bases are, or just the 4 letters that stand for them? All right, yeah, we're getting some answers here: A, T, C, and G. And which ones bind together?
How about what does A bind to?
Oh, that's probably a better question. A binds to T. Yep. And then which base will bind to G? C. All right, AT and CG. And then what is going to hold the bases together? So like, what's the backbone of our DNA structure made of? Yes, sugar phosphate. So we have a sugar phosphate backbone holding together all of these bases. And so if we're looking at just one side, we have a long string of nucleotides. And then when it binds to the matching nucleotides, it forms our double helix. All right, so we've got another poll here.
All right. Let me get to this one here. So this is sort of a challenging one. It's probably our most challenging poll so far. What is the sequence of the red strand? So take your time and use information from what Charlie just talked about. I'll share our results here. So what is the sequence of the red strand? And Charlie, most said C. Is that our correct answer?
That is our correct answer. So if we're looking at this strand across the bottom, we have an A, which will bind to a T, then a T, which will bind to an A, G will bind to C, our next T will bind to an A, C to G, G to C, then we have 3 T's in a row, a C and an A. So yeah, that our third one there is the correct answer. And just for, we'll do one more practice with this. So what's going to be our sequence of the purple strand here?
Okay, so I just launched this poll as well. Please go ahead and answer this one.
While that's running, it looks like we've got a question for Charlie in chat. Why do some bases combine with the other bases?
Yeah, so this is based on the molecular structure of these bases. And so because of the atoms that like make them up, they're going to have these hydrogen bonds so that they'll stick together. And so we have 2 types of bases, purines and pyrimidines, I think is how you say that. And so one of them has 2 hydrogen bonds and one has 3 hydrogen bonds. And so just because of the structure of these different bases, they only have like specific ways that they can link across from their backbones to stick together.
All right, thank you, Charlie. So we're going to go ahead and end this one right now. And my computer's thinking about it.
All right, so it looks like most of you had the first sequence, and that is the correct answer. So if we're looking at these, we know that T binds with A, so we've got an A and then a G, C, A, G, T, G, C, double A, and then a final C. Yeah, awesome. All right, so good job to everyone who got those.
Nicely done.
All right, so then we can use these base pairs to like design sequences that are going to stick together in certain ways. And so we have this thing called crossovers, where if we, I'm going to just try and annotate on here. If we have, instead of this half of the strand having a sequence that would bind to the red one, we have it binding to the blue one instead. So we design these sequences so that they'll bind to different strands. We can actually get crossovers, which are shown here, so that if you have something like this, then you have 2 double helixes that are basically stuck right next to each other because these different strands are attaching to different sequences.
So then, Charlie, in this example here, is the one on the right an example of DNA that would be partially unzipped? Is that how that works?
Um,
And then sort of matched with an alternating or an alternate strand?
Sort of, yeah. So we have two different sequences. And so we've got our blue sequence and then our red sequence. And so if we have instead of strands that match completely, we have strands that only match halfway, and so they'll bind to the other sequences that do match them.
I see. Alrighty.
Ooh, my
And
mouse
then,
is weird.
how can we use base pair bonding in engineering?
Sorry, my mouse is gone. I'm not sure how to get it back here.
Oh no.
I'm gonna try stopping share quick just to,
All right. So
there it is. Okay. Sorry about that. Okay, there we go. Yeah, so in engineering, we can use these crossovers to design sequences that will build structures. So instead of just having these single double helices, we're going to design sequences that will cross over and stick together to bind different helices together in different ways, so we can actually build structures that are really tiny because they're the size of DNA. And so we can use these properties to engineer nanostructures.
oh, neat. And how stiff is DNA? So we're gonna have that poll right now. And this is actually going to be our last poll for the day before we break out into our breakout sessions. So what is the stiffness of DNA? Is it like steel, like hard plastic, or like spaghetti? Is that cooked or uncooked spaghetti?
Or cooked spaghetti.
Cooked spaghetti, okay.
I guess those are pretty different.
And Charlie, looks like we've got a question in chat. What would the crossovers do inside the body or mean for the body? Or do they occur in the body?
So they're not generally happening in the body unless like cells are like replicating and that sort of thing. But we are using this property for engineering and not specifically looking at it like biologically. We're just using the biological principles to design nanostructures.
Okay. All right, so we are going to end that poll and I'll share the results.
All right, so we had a tie between spaghetti and hard plastic. So B, hard plastic, is actually the correct answer here. So when we have these double helices, it actually is going to be like a hard plastic when we're looking at these short lengths of DNA. And so when we have single strands, when the DNA is unzipped and we only have one side, that might be more like spaghetti because it's loose and floppy. But as soon as we have two strands, single strands binding together, so we have a double helix, that's going to behave more like a hard plastic.
Okay, and how are these DNA origami structures made?
Yeah, so using all of these things that we know about base pairing, we can essentially take one long single-stranded piece of DNA, and if we know the sequence of it, then we can design staple strands that will bind it together in specific shapes. So we're going to use shorter strands to form crossovers in different places to make the scaffold strand into the shape that we want. And so we can build shapes like this smiley face or nanoarrays by like attaching different things to the DNA, or even 3D nanostructures out of the DNA if we fold our scaffold into a 3D shape instead of just a flat 2D shape.
And you all are sort of going to get the opportunity to try this out in just a couple of minutes. That's going to be in one of our breakout sessions where you will be working on scaffold and staple strands with pipe cleaners.
Yeah. And so this slide here just shows kind of the manufacturing process of this, how we actually make these structures. And so you first like think of what shape you want to make, figure out how the scaffold, since we have to have our one scaffold going through the entire thing, we figure out where is this scaffold going to go, like what path is it going to follow that it can make this shape. Then we figure out how to design the staples so that they can hold the scaffold in that shape. And then generally we just send an order to a company that makes these sequences for us. So there are other people who do the biological parts and actually make the sequences. So in the lab that I work in, we just do all of the designing. We order our sequences and then we make the structures. So we take these sequences that we have and put them all into solution together. And then we put them through a thermal cycle ramp, which since we have all of these bases, all these different sequences, strands, all in solution together, you might have a lot of things like binding where they're not supposed to. And so we get it really hot so that they all break apart and then slowly cool it down and then like reheat it and cool it and reheat it and cool it. And eventually they all bind the way that they're supposed to. So you won't have just like random A's and T's binding. You'll have like these full sequences binding, since we'll heat it up enough to break, just one or two bases, but not enough to break a whole strand. So as we like heat it and then cool it over and over, we eventually get these structures.
All right. So DNA, as we know, is pretty tiny. So now we're going to talk a little bit about the nanoscale and how small are these structures that we're talking about.
Yeah, so DNA itself is about 1 million times smaller than a raindrop. So looking at this image here, we can see that a raindrop is about 2.5 millimeters across, whereas a bacterium is 2.5 microns long. And then DNA, which is inside every cell, including bacteria, is 2.5 nanometers across. So it's really small. And so just for an example of how we think about these structures that we're making, if you look at one strand of your hair, imagine a football field that's the width of your hair. And the structures that we're designing are about the size of the football. So really small.
Very, very tiny by comparison.
Yeah, very small. And so when we're making these structures, so basically what we've talked about so far is just designing the structure itself, like these different shapes, but we can actually add a lot of different things onto DNA, which is part of what makes it so cool. So we can like attach different optical molecules, so we can attach like fluorophores, so we can look at how these structures are behaving. We can change the...
What is a fluorophore?
Yeah, sorry, a fluorophore is just a molecule that will basically light up when you shine a laser on it, so it fluoresces. Yeah, so when you shine a laser of the right light frequency on it, so that it's you're exciting it in the right way, it'll fluoresce and give off light, and you can use it to signal different things. You can also attach different proteins or design your structures so that they will attach to proteins. So if you're looking at doing some sort of cell study where you want to send different structures into your cells, you could design them so that they will attach to different proteins or molecules on your cells.
All right. And we have one question here. Is when they fluoresce, is that like fluorescent lights?
Yeah, so it's just giving off like one wavelength of light, basically, or a small range of wavelengths. So like fluorescent lights that's generally like one color, right? So that's basically what we're talking about, but like really tiny.
And Dr. Webster Wood has posted something in our chat as well. Would you like to share more about that?
Oh, I was just one example of fluorescence that you might see in biology is there are actually these jellyfish that if you shine the right color light on them, they light up. And that's actually where one of the first fluorophores came from, is they isolated this material, this protein called green fluorescent protein from jellyfish. And so we can use the exact same principle that the jellyfish use, but instead of being in the jellyfish, we can attach that protein to these structures and get them to glow green when we shine light on them. It's a lot of fun.
All right, thank you for that additional explanation. That was really cool. So Charlie, can we make the nanostructures do anything other than just making cool shapes?
Yeah, so that's this stuff. Just attaching different things to our structures to get them to perform in different ways.
Okay. And what sort of structures are you working on in designing?
So what I'm working on specifically is shear stress sensors. So shear stress is the stress that acts tangential to a surface. So if I have a flat surface, the force that would like pushing along the top, that's a shear force. Whereas, other types of forces are like pushing against each other. A shear force is like a sliding force. And so to measure this at the nanoscale, we're designing these structures that would attach to a surface like a cell, or for right now, just, a glass cover slip. But eventually they can attach to cells. And then when you have fluid flow over the top, like water or blood or some other fluid, it causes this sort of dragging, sliding force at the surface. And so it's going to pull on the structure and cause it to deform. And so since abnormal shear stresses in the body, like in your veins, can cause things like atherosclerosis, or like if the shear stress is really high, it can cause platelets to activate. And so platelets activating when you have like a cut or something means that they clot and stop you from bleeding, which is a good thing. But if you just have really high shear stresses, it can cause the platelets to clot when you don't want them to clot, and can cause things like heart attacks. And so being able to measure these shear stresses and see how they affect cells in different ways is really important for solving some of these health problems and figuring out how the cells are going to be affected. So by designing these structures that will deform under fluid flow, we can start to measure these stresses at a really small scale that people haven't really been able to measure before.
Interesting. And we do have some questions in our chat right now. And it looks like one of our mentors was actually answering as well. And
right.
You sort of already answered the one question, how would the cells be exposed to shear stress?
Yeah, so basically any cells that have fluid flow over the top are undergoing some sort of shear stress. So like your veins veins and capillaries, they're all like lined with these endothelial cells that are constantly undergoing fluid flow. Any of your blood cells and platelets and anything that's like in the blood is also undergoing shear stress because it's like in the fluid. And so basically anything that you have fluid flow over is going to undergo some of this stress. So also like in your lymphatic vessels, there's also like you have your lymph fluid instead, and that's also causing shear stress. Your heart undergoes a lot of shear stress because you have blood flowing through there. So yeah, basically any fluid flow is going to cause some sort of shear stress.
Okay, thanks for answering that. So our next part, we can't do it in person, but we are going to be taking a tour of the lab, and specifically of the lab where Charlie works. So Charlie, if you want to get that opened up in the background maybe first, and then you'll be able to start sharing our 360 tour.
All right, yeah, so next we're going to be getting into these hands-on activities where you'll get to do some DNA origami modeling on your own. And so this here is just like some of the first DNA origami structures that were actually made. And so as you can see, like they're pretty simple, right? Like we have these two-dimensional structures, a star, a smiley face, and then these triangles. So they're not super complicated, but it actually was like kind of a big deal when they were first made. Like you can make structures out of DNA. And so we're going to also be making just some like simple structures. We'll be getting into breakout groups for these after the break. So first we're going to be doing a pipe cleaner activity, which will be explained more in a little bit, but basically we're going to be taking a long scaffold strand and then designing staple strands to hold it together in the shape that we want. And so what we can see, like some of these ideas for what to make, like a tweezers, you know, we can make something that will open and close if we move it. But if we design the staples in the wrong way, so it's still held together here, but it doesn't move the right way. So as you can see, like the design aspect of this and how you make your staples to hold things together can really affect how your structure is going to work.
And Charlie, can you show those tweezers again? Those were really neat.
Yeah, so it's just, you know, a diamond structure and then two arms. And so if we pinch it open and close, we can open and close the end. But again, if we don't have the staples designed the right way, so now instead our staples are following this zigzag pattern, and so it's still held in shape, but it doesn't function right. So part of this is just thinking about how we might design our staple strands to make our structures do what we want them to.
Okay. So that's going to be our scaffold and staple strands activity. And then there's going to be one activity after that as well. It's going to be our DNA origami box.
Yeah.
So Peter had a question about it. What would you use DNA structures for in real life? So the DNA origami box is one way that you can potentially use these structures in real life. And you're going to learn more about that in our breakout sessions. So Charlie, if you want to say a couple things about this activity as well.
Yeah, so yeah, these DNA origami boxes are made by, you know, you design a structure in 2D first, and then when you put everything in solution, you have them fold up into a 3-dimensional shape. But I'm just going to go back a couple slides here. So when we're making these designs, they're going to look something like this, right? Like they're all going to be flat, and you're just designing where the staples will go. And so when we have something like our origami box here that we're going to be doing, you know, we have all of these double helices on here showing where the strands will be going, but it's in 2D and it will eventually fold up to be in 3D. So we're going to make it into a 3-dimensional shape from our 2D design.
Exactly. Okay. So in just a moment, we're going to take a quick 5-minute break before we go into our breakout rooms. And once we get there, this is where you'll need all your materials that have been previously sent to you. So again, you will need the DNA Origami box activity sheet. You will need the scaffold and staple strands activity sheet. You will need your 6 pipe cleaners. And then you will also need a pair of scissors, some Scotch tape. And if you have it, you can also use a 3-hole punch for the DNA Origami box activity as well, but it's not required. You can just use scissors or even a pencil to poke out the white holes. So with that, and when we start our breakout sessions, we're going to start with the staple strands activities first. Our mentors are going to guide everybody through those. We're going to have randomly assigned breakout sessions. So we'll do the scaffold and staple strands activity first, followed by the DNA Origami box activity. And then after that, we'll reconvene, we'll come back, we'll have a short Q&A session for any of our lingering questions. And then we will close our sessions for the day. So with that, right now I have on my clock 11:29. So at 11:35, we will go into our breakout rooms. And I will be creating our rooms while we're on our break here. Okay, so welcome back, everybody. I hope you enjoyed the hands-on activities. And again, if you weren't able to finish, the how-to videos are going to be posted on the Canvas site sometime next week, along with additional resources for you to use so that you can learn more about DNA origami. So I'm going to go ahead and share my screen. Okay. So raise your hand if you can see the PowerPoint presentation where it says conclusion. Great. It looks like everybody is able to see that now. Okay.
Kristen, I think we have a question about what the recording is going to be used for.
So yes, so we are going to be sharing that first on the Canvas site, and then it will also be shared on the Gelfand Center website later on. So we'll have access to that, and we'll be able to let friends that weren't able to participate today be able to see these recorded sessions and do the activities on their own. So I'm going to turn on screen sharing here for just a minute. And I'm going to stop sharing. Does anybody want to share their creations, their scaffolds, or their boxes?
I can.
Okay. So Allison. You should be able to show your video.
I am.
Oh, there you go.
So I didn't get to finish my box yet, but with the scaffolding, I guess I made like a basket with a hole in it. I guess I didn't really know what to make.
All right. Thank you, Allison.
I could share next. I made the DNA box.
There you go. All right, nice.
I can share next.
Go ahead.
So I tried making the pyramid. It kind of got a little squished, but.
Oh, I see it. There you go. And then Mira.
I'll go next, yeah. I tried to make the triangle, but then it turned out to be a circle.
All right, thank you for sharing. Cool.
I could share next.
Go ahead.
I made a little necklace for my chicken.
Okay.
Beautiful.
All right. Nice job, everybody. Okay. So I'm gonna
Looks like in the chat, Peter wanted to share too.
oh, we missed one. Okay, hold on one second. Peter G or Peter D? Peter D. All right, one second here, Peter. Find you.
Looks like he's got his camera and mic on.
Oh, okay. Oh, there you go.
So I made the tweezers, and surprisingly, they actually work.
Nice.
So, and then I made the DNA box to hold all the extra parts.
There you go. All right, thanks for sharing. Okay. So I'm gonna go back to the PowerPoint here. So now, does anybody have any questions? So raise your hand if you have a question, and then we'll call on you. And we have just a moment here. So how about Risa?
Hi, okay. So we were in the breakout groups and we were talking about medicine delivery, and like you said, they code the DNA, and I was just wondering how you do that, how you code DNA.
Yeah, so like, do you mean like how to make the specific sequences?
Yeah.
Or yeah, so I'm not an expert in this because basically like what my job is to design the structures we want to make, and then I order the specific sequences that we want. But I know there's a process where they make these sequences in bacteria, and because you can make the bacteria replicate pretty quickly, so you can make a whole bunch of DNA at once. And there's another process called, I think it's PCR, to pull out the specific sequences that you want. And so I'm not an expert in how they do that, and I don't know a lot about it. But yeah, we just, you know, pick the sequences that we want and that are going to be useful. And then we have, because we're just engineers, and so then the biologists will actually make these sequences for us using like bacteria and other things.
Okay, thank you, Charlie. All right, so we're gonna go ahead and stop with our questions today so that everybody can get out of here by 12:30 as promised. So thank you everyone for joining today. And special thanks to our presenter today, Charlie. So I'll give her a round of applause here. And thanks also to our mentors and to my friends that have helped coordinate this program today. I hope you've enjoyed it. I will send out a survey to see what everyone thought about our program, and you'll probably receive that via Canvas. And then stay tuned for future sessions and check back on Canvas next week, and you'll be able to see our recorded session and also our how-to videos that accompany the activities. And I'll also be posting some additional info sheets that accompany our activities as well. And I might ask Charlie for some additional info so that you can sort of extend your learning beyond today if you want to learn more about DNA origami. So yeah, thank you everyone for participating today. And have a great weekend.
Thank you.
Thank you.
Thank you. Bye.
Thank you. Thanks. Bye.
Thank you, Charlie. Bye.
Thank you. Bye-bye.